Hello, my name is Matthew Dawson, and this is my project, Pilot Screening to Identify Social Circuitry. So a little bit of background. It has been shown that zebrafish are highly social animals, which makes them a great model organism for studying social behaviors. The neuronal circuitry that underlies social behaviors continues to be enigmatic and difficult to model. Through selective ablation of neurons, you can find populations of interest that are necessary for performing social behavior. And lastly, knowing this circuitry will allow for more informed interventions as well as hypothesis testing. And so the hypothesis of my study is that neuronal circuitry underlying social behavior can be deduced through ablation of unique expression patterns. And the way we go about doing this is we first generate a cross of two fish. The offspring from this cross will have either green fluorescent protein or red fluorescent protein. And what this tells us is that the fish with the red fluorescent protein have a genetic insertion that allows for induced cell death. And we induce this cell death using a fairly non-toxic antibiotic. Once this chemo-genetic ablation has been induced, we'll take fish up to the virtual social assay. And in this assay, fish are placed into a watch glass, and a dot is projected into the watch glass. And fish interact with this dot as if it's another fish. And what zebrafish like to do is swim really close to each other. And so from the distance between the fish and the dot, we can calculate this social index, which is the x-axis of my top graphs. And then we can see if ablated fish are more or less social than non-ablated fish. And lastly, after the fish go through the virtual social assay, we extract samples for confocal microscopy, which is what you see pictured here. So these are 4 different lines that were tested, and I have forebrain images from all those lines, and the forebrain is where this expression pattern is localized. And what was found was that lines 1 through 3, although they were not clearly exhibiting social differences, these expression patterns can still be beneficial in describing and recognizing expression patterns that are not necessary for social behavior. But I did find one line 4, where ablated fish showed clear deficits in social performance. And additionally, the average speed was also higher than non-ablated fish from line 4. To build upon this, some future directions, it would be beneficial to enhance these images with antibody tagging and increased image size, which will allow for better modeling of the circuitry. And also using image processing to begin mapping these unique populations onto an average brain, which will allow for a visualization of what this circuitry may look like. Lastly, further analysis on fish that did not clearly exhibit deficits in social behavior to see if there were any more nuanced differences that the eye can't pick up. I would like to close by just thanking the McNair Scholars Program, as well as the Washburn and Eisen Lab from the Institute of Neuroscience for all their support, and thanks.